It's my great pleasure to invite Professor Hongyu Zhao from Yale University to give a IAS talk here. Before his talk, let me briefly introduce our speaker. Hongyu is the ILV Hiscock Professor of Biostatistics, Professor of Genetics, and Professor of Statistics and Data Science. Hongyu is now Department Chair of Biostatistics of Yale University, and Hongyu is now serving as a co-editor-in-chief of JASA, which is the highest level of journal in the statistics. Hongyu's research interests are the applications of statistical methods for molecular biology, genetics, drug development, and personalized medicine. Hongyu has received a lot of awards for his contribution in statistics and public health. Among them, Hongyu received the Mortimer Spiegelman Awards in 2008, which honors the top statistician under age 40 for his contribution in public health statistics. And now nearly 10 years passed, but Hongyu still looks at the age of 40. So far, Hongyu has trained more than 70 PhD students and postdocs. Many of them are holding tenured or tenure-track position in many different major universities around all over the world. And as the supervisor, I think Hongyu is always been very supportive. I think that's the most important thing I learned from Hongyu. Try to be supportive to the student. Now let's welcome our speaker to give this talk, Dissecting Genetic Architecture of Complex Disease by Genome-wide Association Study. Thank you.
Thank you for a very kind and generous introduction. It's always great to come back to Hong Kong. A lot of friends here. I'm looking forward for the party tonight. Today I'm going to share some of our recent work analyzing genome-wide association study data as a way to better understand the common disease mechanisms. And I was told this is a general talk, so there's going to see very few equations. There's a lot of words, some pictures, and hopefully by the end of my talk, everyone's having a good time. So this is the outline of my talk. So for those of you who are not really familiar with the acronym GWAS, so I'll spend 5 minutes to introduce what GWAS is. And then I'll talk about how statistical thinking and modeling can be helpful to really understand the information in the data set. That will be quite different from you may have learned in traditional linear regression models and other standard coursework. And then we'll delve into some of our computational efforts to really understand the functional regions in the genome and how these annotations actually can help to shed light on the disease mechanisms for a variety of diseases, both in terms of the tissue cell type relevance as well as the genetic correlations among a number of diseases. And also give you an example about, it's a fun exercise we did recently to think about, we always teach students binarization, decimalization is not a good thing. I'll give you some example that may be a good thing sometimes to really look at the genetic the mechanisms. And I'll lastly just more entertaining part about how this data can really help you to understand individual risks for different complex diseases. So this is a rough outline of my presentation. Just feel free to stop me anytime. So I don't need to cover every single piece here. So you may or may not recognize what's on the top left, right? That's how the human chromosomes will look like or considered 60 years ago. When people still having trouble to count how many pairs of chromosomes actually people had. 60 years later, we know a lot about much more than we did many years ago. So this back 16 years. There's two concurrent efforts, both aim to identify or sequence of human genome is published, and side by side, or one in Nature and one in Science. And that's how was the output from these two papers. So essentially, it's compiled in a series of books, right? That's how you can really look at the sequence data, right? That's really, you can go into a library and borrow these books. You can look through it, and for volume 1, volume 2, volume 3, you can open a page, and that's what you're going to read. It's a long sequence of ATGC. So that's the problem we are dealing with. So actually, we are given every single person this, now with the technology, this large collection of volumes of books, and our goal is to really understand what's the meaning of every single alphabet, every single alphabet in this book. So that's a challenge. And first, among the most, if you have every single one's books lining up, for the majority of the part, when you open most pages, your book and my book will be identical, right? But from time to time, our books will differ certain positions. So 2 years after the sequence, the human genome, people started on international efforts to really identify where the people's books differ at what positions, right? It's so-called International HapMap Project, including 270 individuals around the world, 90 from Africa, 90 from Europe, 45 from Beijing, and 45 from Tokyo. So they use these people's genotype data and to compile a collection of these variations to really identify how much we differ, where we differ. And there's a series of papers published 2, 4, and 7 years after launch of the process. We get a deeper understanding of the variations among ourselves. And with the sequencing technology, people launched a so-called 1,000 Genomes Project, where you can sequence a whole human genome. And that means that we can really compile a book for every single one in this room. Okay? I said for most part, 99.9%, we are the same. For the part that we are differ, there are maybe different ways we can differ from each other. The most common polymorphism is called single nucleotide polymorphism. That's when these two chromosomes, they are identical at 4 nucleotides, but differ at one particular one. So these C and G, they are called single nucleotide polymorphism. It's a SNP. Okay. And when we open our books, that's where we differ these bases, and we hope that we can find the basis that can really may have some implication for our phenotypes: height, hair color, obesity, cancer risk. Right? That's a goal. We have all the information here. How we can really use that information to identify the genetic basis, what genetic variations actually can give us higher risk for different cancers, Alzheimer's disease, schizophrenia, etc. And there's a very reliable and also very effective, cost-effective platforms to really you don't need to print every single word now. You can just get a selected field to understand human genetic variations. So these chips here in the middle, Omni 2.5, on this chip, actually there's a 2.5 million sites in the genomes that are being probed. That means instead of having a book with 3 billion letters, this one actually going to look at 2.5 million. It's about 1 out of 1,000 it's only 0.1%, but they actually capture a large variation of the human genome variations here, 2.5, and these used to cost a couple $100 to get this chip genotyped. And now the most national or international consortium, they use like less dense chips with about 700,000 sites, not 2.5, and they cost like $20, $30. It's much cheaper. Yeah, they're quite reliable. So we have this technology now. We can go to every single one in this room, and we can genotype, and then we can use this technology to ask questions about what genes, what variants are associated with different diseases. So this is a very simple study design. If you want to understand what variations may be associated with a given disease for breast cancer or prostate cancer, you just get DNA, these materials from people with the disease, and the matched controls without the disease. And we can get data like this, right? So we have a lot of people here. Each row corresponds to a particular individual. So in this case, the first column represents the phenotype that for a disease. 1 means someone has a disease, 0 means someone doesn't have the disease. So this is the phenotype. And our goal is to identify out of millions of columns here, every column represents a genotype or single nucleotide polymorphism. There may be hundreds of thousands or millions of columns here. We want to understand which columns are associated with the response variable Y. That's how it's taught in linear regression course. So we can look at array, we can do univariate regression, we can do very simple statistics to identify which ones of those columns are associated with the trait. So that's essentially the very simple summary of what genome-wide association study design is. It's very straightforward. You want to study a disease, you go out, collect people with the disease phenotype and people without the disease, and you get their genotype, and you do a lot of association tests. Suppose you get millions of markers, and you calculate, you correlate the SNP genotype for every single column with the phenotype, and you calculate p-value. So how to calculate p-value? This is an example. It's from step 101, right? Suppose now we have collected genotype data from 200 cases and 200 controls. So this is one of the maybe a million columns. Suppose at this column, we have 3 genotypes, AA, AB, BB. And among 200 cases, we saw 72 people with AA, 96 with AB, and 32 with the BB. Controls, we have 50 AA, 100 AB, and 50 BB. Okay? So we want to test, is there any association between the phenotype and the genotype, right? What we do standard chi-square test, right? Everyone knows how to do chi-square test. This is observed, and this is expected if you assume the rows and columns are independent. And this simple R code, and you just write it down, and you got p-value 0.018, right? So you do this for marker 1, and do this for marker 2, marker 3, etc. And the only difference between the standard chi-square test and the GWAS setting is because we are really testing for millions of markers. So this one, if you do a single test, this p-value will be considered maybe okay, right, to make a statement about statistical significance. But the fact that you are really testing millions of markers, and the chance seeing this p-value is going to be not that small among those millions of markers. So people use very stringent threshold. For example, 5 times to 10 to the minus 8. The reason the number is chosen is if we test 1 million markers roughly, if you use Buffroni correction, so that gives you genome-wide false positive control about 0.05. That gets back to Fisher's proposal many, many years ago, right? So just a very, very rough approximation here. Okay, so far so good. And so suppose this is a hypothetical example that illustrates a lot of some real data results. So suppose you are interested in a particular phenotype, say for example breast cancer, and you type 1 million markers, and you do 1 million tests. And then you can do count. You can count how many markers they have a p-value less than 10 to the minus 8. Okay? You have 2 markers. Like 12 years ago, 10 years ago, these 2 markers will get you a paper in Nature or Science. Right? That's because it's below 5 times 10 to the minus 8, this magic number. Okay? But still, there's 4 markers. It's between 10 to the minus 7, minus 8. So these 2, they may or may not have made it 5 times 10 to the minus 8, but we can only say much about it, right? Now if you look at 10 to the minus 6, now we have 7 markers that have p-value less than 10 to the minus 6. If you want to have a paper say in Nature Science, you cannot make a claim about these 7 markers being associated because you don't have the evidence, statistical evidence that's cut 5 times 10 to the minus 8. But you know what? If all the hypothesis tests I'm doing are right, the chance of having p-value 10 to minus 8, if there's no association at all between any one of these million markers and the phenotype, it's 10 to minus 6, right? If I have 1 million markers, on average, I only expect to see 1, but I see 7. Okay? Now if you go down one more, 10 to minus 5, now I see 20 here. If there's no association at all between any one of the million markers and the phenotype, I expect to see 10. 1 million times 10 to minus 5 is 10. I see 20. If you go down 10 to minus 4, I can keep doing this, but 10 to minus 4 is 180 versus 100. So what do I know? So I say, okay, there's 180 here. If there's no association at all, I expect to see 100. So roughly, maybe 80 out of this 100, they may be associated with a phenotype. But just I don't really know which 80 of these 180, right? So we think about the rough, very like a superficial definition on FDR. The false positive rate here, if I make a claim is 180, would be 100 over 180, right? That's the false positive rate in this set. But I don't really know which ones, right? So if just focus on writing a paper that meets a threshold, all the information here is lost. You don't report anything below this. And what am I trying to tell you or to discuss today? Actually, there's a lot of information in this, the lower rows here. You can rescue through some statistical methods and also coupled with some functional annotation efforts. So this one is still probably most recent published GWAS, looking at how many, look at the genetic association with height. So in this study, they analyzed the height data from about 250,000 people. They identified about 700 variants at genome-wide significant. Together, that's how many identify. So it's a lot, right? And then this is a study published also like more than 3 years ago, looking at the genetic basis for schizophrenia. And in this study, by studying 37,000 schizophrenia cases versus 113,000 controls, they were able to identify 108 regions in the genome. So it's a lot of regions have been identified to be associated with the schizophrenia. And every single variant here actually met the cutoff, significant cutoff, 5 times 10 to the minus 8. So there's no ambiguity about statistical significance, and also have been replicated. And now I can get the question of how this can tell us about the genetic architecture. So I will explain what genetic architecture means. It's a very, very general, loose term. For example, I can ask question of how much heritability can be explained by the identified variants, right? So we have 697 variants that have been identified to be associated with the height. Now, if I want to use these 697 variants to predict someone's height, how well we can do, right? So this is what the first question is. So for example, in the abstract, I think they already gave the results here. Right? We show that the most strongly associates 2,000, not just only 700, but 2,000. They can build a prediction model of someone's height using the top 2,000 variants. If you look at how well the model prediction is compared to the real height of an individual, the R square is about 0.21, right? If you increase that to 3,700, you get R square 0.24 and 0.29. So this is what you can really do to really answer that question. That's standard, not so standard, but still standard in a way that you build a prediction model using the top ranked predictors you identify by applying simple threshold cutoff. And because now you have 250,000 people, you can afford to estimate a few hundred or a few thousand parameters. So that's possible. And you build that model, and you can see, you can try to understand how well that model does, right? So that's what they did in this paper. And the other question you can ask, how much heritability can be explained by the studied SNPs? So that's like not a typical question you would run into in statistical courses. So this question is the following. Suppose I have this genotyping array with 1 million SNPs. So what this question is really asking is, if we could identify every single SNP that actually has an impact on the height if we then build a model using those SNPs, how well we can do the prediction, right? So this is question is whether we're able to even answer this question, because first it's be impossible for us to make a statement, now I have identified every single variant on this chip, this microarray that's associated with height. There's any way we can get around the problem. Yeah, so going back to the historical side, it's like this is a paper actually got a lot of interest. This is the first one starting this so-called missing heritability. So I can say you go backward, not 2014, but 2008, 6 years earlier than 2014, with this study about 250,000 people. So in this study, the authors actually analyzed the genotype data from about 25,000 Iceland people, 3,000 Dutch, about 17 or 1,800 European Americans, and some African Americans. Because of the smaller sample size, they didn't identify all these 697 variants, but rather they only identified 27. But still, at that time, it's a pretty good large number, 27 variants associated with the height. And then they built a prediction model for someone's height using these 27 predictors. And they got R-squared like 4%. So this is a paper about. And so people, you may be happy, maybe upset. A lot of people were very upset by the result. Why is that? Because height is probably one of the most heritable traits in humankind. And it's about 80% people estimate by looking at the parents' height and children's height. And when they spend a lot of money, put in a lot of efforts, they identified 27 regions, they were only able to explain 4% heritability. That's only 5%. So the question was, how large is something you need to do, how much more money to spend? And there's a lot of controversy in this, after this. People say, okay, we are now really looking at the wrong targets. We should be looking at rare variants. Because the genotyping array is only genotype common variants. These are the single nucleotide polymorphisms that you see maybe in 1% or 5% of the population. And some people argue, this is a total failure of a common variant hypothesis. You should really go to the rare variants. And I'll say you should even go beyond single nucleotide polymorphism. You go to a copy number variation, much larger structural variations. And also these people can make things even worse, saying we should consider much more complex models. These genes, they don't act in isolation with each other. They should like gene-gene interactions, SNP interactions. So it's a lot of confusion about what the next step is for genome-wide association studies. Hi.
What techniques do you use in this paper? What method?
Regression.
Just linear regression?
Just linear regression, yeah. Very simple linear regression, yeah. Okay. So that's about 2008. And there's editorial written in Nature Genetics about the missing heritability. And a lot of people in the field were confused. Also, even there's a New York Times article and really criticizing or like being very pessimistic about the future for genome-wide association studies. So that's about 2008. And then 2 years later, I think this is a really landmark seminal paper in the whole GWAS, in the literature, from Australia. So they try to look at height, right? So what they did is instead of looking at land population, they have their own population. Look at about 3,000. Actually, many fewer than the other study, right? And so look at the difference here. They show that 45% variance can be explained by considering all SNPs simultaneously. So that's the major conclusion in this paper, saying that now I have a study with about only 3,000 SNPs. What these authors, what they claim is the following. If they could identify every single variant out of these 300,000 of fighting height, they could build a prediction model that can predict the height with R-squared 0.45, a tenfold increase was people reported earlier. So this is the big gap between 4% and 45%. If you believe their story here, then all these other arguments about rare variants, gene-gene interactions, and copy number variants, other things, they are become less relevant because as even you only have 300,000 SNPs with a relative sample size, actually this microarray chip is able to capture 45% out of 80%. So it's a pretty big proportion here, right? So what's the difference between these two numbers? And this actually found is a maybe good story for statisticians. Say, look, we have some tools that can really help you better interpret your experimental results. So the model setup is no more complex than this following, right? So let's assume we have this trait model, Y, and the trait height is affected by the additive effects of a lot of SNPs. For example, we have 1 million SNPs. We assume your height is a function of these 100 million SNPs. But these 100 million SNPs act in an additive fashion, right? So this xi denotes the genotype, whether you have 0, 1, or 2 copies of the rare variants or the less common variants. So you have this linear regression model Y with about 1 million terms. And beta quantifies the effect size of this genotype on height. Okay, so there's nothing more complex than this very simple additive model. The only difference is that we have 1 million predictors. Even in the case about 2014 paper, we have 250,000 individuals. We are still only 1/4 of the sample size, only 1/4 of the number of parameters. So clearly, you will not be able to estimate every single beta here, right? It's much more parameterized than the sample size. So what do we do? But just take the model here as a very simple one. So what we're trying to really estimate, or the question is asking the following: how much variance can be explained by all the SNPs on the chip? It's asked the following question. This is on the left-hand side, the variance of Y. This is the variance for height, right? So this corresponds to the variance explained by all the additive effects of SNPs on the microarray. And this is the additional noise. So the second question we are asking, how much variance can be explained by all the SNPs on the microarray? Essentially, it's a ratio of this term over variance Y. So that's the question. So the real problem now becomes whether we'll be able to estimate the ratio of these two terms, right? Without really identifying every single SNP affecting height. Okay, so this is a question. And it turns out that's possible by just assuming that instead of aiming to estimate every single variance effect, we assume this beta is a random effect. All these beta, we don't want to really, we are not aiming to estimate every single beta. We just want to estimate this beta. We assume this beta comes from, say, for example, simplified normal distribution with a mean 0 and some variance. If we make that assumption, it means that that's a big assumption, right? That how that assumption can be true. Let's assume that we make that assumption. And then this model only has one parameter here, beta with a mean 0 and a variance, sigma square. And then this model becomes solvable, right? So it's a basic idea. And this is a standard REML approach. Actually, when this model is applied, assuming it's random effects model, and so this is a little bit later paper. So for height, this sample size, the different sample, about 12,000 samples. And this is already estimated about 45%. And the heritability 80 to 90%. So actually, more like the same consistent with the reported from Australia. If you do the GWAS, collecting all of the SNP associated with the height, you only get R squared like 10%. So in this case, this is pretty much consistent with what's reported by Peter Wischer and colleagues. So after this paper is published, and the same random effects model approach has been applied to all sorts of diseases, and many papers have been published. And but one thing that really puzzled us is that in this random effects model, we assume that the beta for all different SNPs, they actually come from normal distribution with a mean 0 and the variance is sigma square. That means this model treats all the SNPs equally, right? If I have 1 million SNPs, this model assumes all the SNPs, they have some impact on height. That's clearly not quite possible. You may say that may be okay, but we think this model may be specified. And so at that time, well, I think the time was at Yale, and I said we were discussing this. I said this cannot be true. So let's do some simulations and to abuse the model, and to see how well their procedure works. So this is a probably result from 10, right? And so we did a very simple model without looking at the 1 million markers, but still like substantial large, is 20,000, right? So we assume a model where Y is strongly determined by the additive effects from 20,000 variables. And 20,000 each beta has a normal distribution. So what this is, this one, 20,000. And then apply this random approach to estimate the variance, to see whether we can estimate the variance of this one divided by the variance of Y. And the model is correct, it's a random effects model, and it turns out actually it's doing a pretty good job, right? So that's fine. But how about only 10% of the SNPs actually have some impact on height, not 100%? So that means 90% of betas are 0. The other 10% follow normal distribution with the mean 0 and variance sigma. Whether we'll be able to do it. Now it's a model, it's different from the random effects model. So that means, look at this one, 2,000 out of 20,000. It's still doing a very good job, okay? So I say, let's push even harder to see how robust this particular approach that can go 20,000, that 10%, how about 1%? 1% is 200, right? So look at 200 here. It's still robust, it's still very good. So what's going on here? Now let's abuse even more. Let's turn to 0.1% or 0.05%. Now it starts a little bit, but even like 0.1%, 1 out of 1,000, suppose only 20 markers out of 20,000, they have some impact on height. If these 20 markers follow normal distribution with mean 0 variance sigma, this standard REML procedure is very robust. That totally surprises us. So we had no clue what's going on, how to really analyze this mathematically. So we went to a guru on REML and Jiming Jiang, and so he's written books about random effects models. Actually, we figure out actually there's some mathematical, theoretical reason why this procedure is very robust. And this is in the sample size, P is the number of markers, M is the number of associated markers, M over P represents the sparsity of the SNPs in terms of the model. We was under assumption, very like a pretty general assumption, as long as N over P, sample size or number of markers goes to a constant, and the number of markers associated with the disease over total number of markers goes to a constant, this procedure is still robust. So that's after a lot of sort of derivations. But just show you, this is kind of interesting example where statistical thinking actually can help you to better interpret the results, not 4%, but 45%. Actually, this gives some people a lot of, didn't waste a lot of time or effort to look for rare variants. That's a big deal. And also give rise some interesting statistical problem that I work out and turns out to be some new theoretical properties about REML, which Jimmy was very pleased because REML is really very robust, even under this extreme setup. So the second topic I want to talk about is, now we have this GWAS data, we analyze this way, and any other information we can bring in as a statistician and a computational biologist to really gain some other insights, not just look at the top signals, but can we understand a little bit more? So the first thing I want to mention that in the previous slide, all the columns, they have the same font size, but now they are different font size, you may notice. That the same trait values, but some columns, they have larger font size than other columns. Why? Because when we're talking about SNPs, they are not just these alphabets in this book, in the library, but rather these SNPs correspond to different locations in the genome. Some of these variants actually disrupt the protein product, these non-synonymous SNPs. And a priori, these SNPs may likely to affect the function of the protein and the function of the biological process. So the larger font here suggests this particular column may just without any compared to the other SNP here, which may reside in the intergenic region where people know little about their function or don't have much evidence for function. Between these two columns here, M1 and MM, if you get the same p-value of 0.001, if we want to only have one replication experiment to do, which one to go after? You want to go after replication experiment, this one, because this is functionally relevant from biological perspective. And you want to put more bet on this one. Now you try to incorporate some prior information into the analysis. So we have been developing tools to integrate different sorts of information, both from such information here in terms of where the SNPs reside, also from experimental studies. For example, the ENCODE is a community effort acronym for Encyclopedia of DNA Elements, where they do a whole bunch of experiments trying to test the functional potential function of different regions in the genome. You can think about the data come out like this, right? I have these all different experiments. For this experiment 1, experiment 2, experiment 3, every single position in the genome, to simplify, there's a score. A higher score suggests this position may be functionally relevant. Its lower score means that's probably not, right? So this is a GURP. We're looking at the functional, the sequence conservation across different species. So if a particular position is highly conserved across between human, mice, monkeys, down all the way to even lower organisms, that may suggest this position is critical, is relevant. So this may have a higher GURP score. We can look at every single position in the human genome, only 3 billion of those. And also we can look at experiments. You can see that based on experiment results, it may suggest the first 3 positions probably are not as relevant as these 3 positions. So each experiment gave us, you think about very simple ways, a score about the functional relevance. And our goal is to integrate the results from different experiments to have a score for the potential functional relevance of every single nucleotide in the human genome. So that's sort of the setup, where we have a lot of experimental results, sort of sequence comparisons, etc. We want to combine all these different pieces of evidence to have a score assigned to a nucleotide to every single base pair in the genome, so that a higher score means functionally relevant, a lower score means no. Hi.
Sorry.
That.
In these different experiments, can you weight the different experiments differently because their impact on function of a SNP might be more or less?
That's a good question. Yeah, we just let them learn themselves. Yeah, they learn themselves. Yeah.
So it can change.
You can change. Yeah. Yeah. So we just built this very simple latent class model where every single nucleotide there's 2 states. Very naive, right? 0 means this site is functionally not relevant. 1 means it's functionally relevant. We just want to combine all this data to infer what's the probability this site is 0 or 1, right? So this is pretty straightforward. And this is just a very simple naive Bayes model. I just keep everything here is simple EM algorithm, but only run 3 billion times roughly. Okay. So these are genome canyon results where we integrate all the ENCODE data sets. So this is more of the visualization how the data look like. So this from left to right, you just go nucleotide 1 base pair at a time. And the height here represents the posterior probability of a particular site being functional, right? You see the chunks here that suggest these regions are potentially functional. There's a valley here, say it's probably potentially not relevant. So we can look at this organism level, say this is a functional annotation for the human genome for a whole body. Let's say we have different body parts. We have eyes, we have hair, we have skin. How come the functional relevant different parts, different body parts will be the same for the same nucleotide? Because different genes get expressed or get depressed or etc. So actually we use data from the roadmap epigenomics project where there's a different cells, different cell types isolated from different body parts, right? So using the same framework, but we can combine the cell types coming from particular organ or tissue here, brain, the digestion system, lung, heart, etc. Right? So we can really build an annotation in addition to the whole organism, but also look at individual tissue type. So this is our illustration, the annotation results. we run like different ones. We do another like 3 billion times the EM algorithm for each update. So it's the brain, GI, lung. And you can see for the same region, hemoglobin region, and the functional annotations, the computational functional annotations are quite different, right? In this case, hemoglobin is, like you're happy to see, is this region is functional relevant in blood, but not so in brain, right? So you do see the tissue-to-tissue variations here. So how this will help us to better understand the genetic architecture or the genetic basis of common diseases. So you can ask questions, for a given disease, which tissue or cell types are relevant? Like I asked for schizophrenia, obesity, etc. How the functional annotation information here can help us to start looking at this question. So the basic idea is very simple, right? So for every single tissue or organ, we have this functional annotation to tell us which regions are likely to be important, which regions are not likely to be important, right? And also for genome-wide association studies, every single SNP, we have a p-value, right? We can look at the p-value, the statistical evidence association between every single base pair and the phenotype. We just look at the overlap between the GWAS hits and the functional rate. So very straightforward, right? So we have you can think about, we have a functional annotation in the genome for blood. Also, we can overlay the GWAS signal, the p-values, over blood. Yeah, if a tissue is relevant, then we expect to see a higher overlap, right? Because you see that the evidence association should mostly come from the functional regions. If the tissue is relevant, this is very straightforward, very simple to understand. And then you just very simple to quantify that. So for example, suppose 10% of the genome in a tissue is functional. This is very naive. But the SNPs in the functional genome actually explain 30% of genetic variance. And now you see a threefold enrichment. So that may suggest this tissue is relevant. And you can have different ways of calculating statistical significance. I'm not going to do details here, but the basic idea is just consider overlap between the GWAS hits and the tissue-specific annotations that we build for different tissue types. And so this is the result that we found. So for schizophrenia, right? So you look at the overlap between schizophrenia, GWAS hits, and the functional regions in brain, GI, lung, heart, blood, muscle, and epithelium. You'll see the largest overlap, the most significant overlap is the brain. So schizophrenia is a brain disease. You do see that actually by combining GWAS results with the functional annotations, you do see that. So that's assuring. And the other one is you see the Crohn's disease, and also ulcerative colitis. These are sort of the two, immune disease. And you see the tissue that is most relevant in terms of statistical significance, actually blood. It's to do with the immune system. Also, it's quite assuring. So that's good, right? So we are seeing the things we expect to see. And initially it was unexpected, but as people repeat over again, is a body mass index, right? Body mass index is, what's body mass? Is your weight in kilograms divided by your height, the square of your height in meters. And the body mass index is really indicates someone's obesity, right? And if you look at the GWAS hits for BMI, and what's the most relevant tissue for body mass index? So I have this here separate for males and females. And as you can see here, the most relevant the tissue type is the brain, not the digestion system. It's more like your appetite. You feel happy about eating things that makes you eat more, and you'll become more obese. And also the second most relevant one in males, epithelial, right? So that's what you want to see. It's kind of, there's some difference between males and females. And also you look at the waist-hip ratio. It's another measure of the body shape, right? The waist-hip ratio is related to the obesity. And the most relevant one for males here is muscle in terms of what's relevance. I mean, when you think about the tissue, or maybe you're fighting the waist-hip ratio for males is muscle, whereas it's a different. The most important one for females is a GI, not the muscle. And so this can be an interesting thing to think about, like if you want to study disease, what tissue, what other things may be relevant. And the other example that, and also we learned later on, there's some kind of people are actually converging on similar observations like Alzheimer's disease. So Alzheimer's disease, everyone knows about Alzheimer's disease, a very serious neurodegenerative disorder affecting a lot of older adults, older people. It's a late-onset Alzheimer. And through genome-wide association studies, many risk loci have been identified and how implied lipid metabolism and the innate immunity, right? So the same thing, we just look at the Alzheimer's disease, the GWAS results, overlap that with different tissues. And the most significant overlap enrichment happens for the immune system. So where's the brain? Brain's here for Alzheimer's disease, right? So brain's didn't pass the statistical significance. And the second one, we also look at the Parkinson's disease. It's another like a serious form of neurodegenerative disorder affecting your brain, the Parkinson's disease. Yeah, again, it's the immune system most relevant, followed by epithelium. The brain here didn't make it at all, right? In terms of the genetic susceptibility for Alzheimer's disease or Parkinson's disease, they don't seem to act through the brain, rather through your immune system. And we look at the cell type level because that's based on aggregating different cell types within a tissue organ. We look at different cell types. There's a consistency between Alzheimer's disease and Parkinson's disease. The most relevant tissue type, the cell type, is a monocyte CD14+. It's another immune cell type. And the other significant one for Parkinson's disease is the liver, to do the lipid metabolism. And look at the brain here. For Alzheimer's disease, for Parkinson, they just seem not relevant at all. And also, we can look at the genetic, or they can look at the functional relevance of different tissues, but also we can use this GWAS data to ask questions, is there any shared genetics between two traits, right? We all know there's people who are obese, maybe tend to have some other diseases. There's any genetic basis for that. So this is a question we can ask for a lot of different pairs of disease. And so this is based on the result we just published in the American Journal of Human Genetics. So the model is very straightforward. Again, you already seen this model. I hope you remember this model. It's a random effects model. Why? It's determined or affected by the additive effects of a lot of SNPs. That's for one trait. Now for two traits, you have two equations, right? Two regression models. It's nothing more than like listing these two equations, right? And what do we mean by genetic correlation? So it's beta I quantifies the genetic effect of the ith SNP on the trait 1. On gamma I quantifies the genetic effect of the SNP I on trait 2. When we say there's no genetic correlation, we sort of probably we mean how much beta I, how much the ith SNP affects Y, the first trait, has nothing to do the effects the same SNP on the second trait, right? So there are probably, we can say, in the statistical term, we can frame as there's no correlation between beta I and gamma I, right? That's no genetic correlation, right? On the other hand, if there's a correlation between beta I and gamma I, and if we can really make statement, the correlation is different from 0. Also, we can even quantify the degree of genetic correlation. The correlation there, we can say these two traits, they have some shared genetic basis. And also, we can use the correlation between beta I and gamma I as a way to quantify the degree of sharing between the two traits, the genetic sharing between two traits. So this is very straightforward. And also, it turns out that not only will be able to estimate the genetic correlation, but also it can be estimated without having access to individual genotype data. So this is why this is important. This is important because when a lot of studies publish their results, they do not release the individual genotype data. There's confidential reasons, other reasons they don't want to release. But summary statistics are much more abundant. You can much more easily have access to summary statistics. And the genetic correlation here, rho I, actually can be estimated with the summary statistics only. Okay? So I will not go into the details here by saying, well, we can do it, and we can do better than some existing methods. So here, the green bar is the previous method. The other bar is our method. You see, pretty much we are unbiased, And also we have a smaller band, and so we're saying we have tighter sort of estimate. So we're doing good job and better job. And also this is sort of the power to identify genetic correlations. This is our power. This is the power for the most commonly used method. So this is a good thing. Now when we apply our method to estimate genetic correlations among a lot of different diseases, and it's about 40 plus diseases. So this is the summary plot. So in this plot, the darker blue means higher positive genetic correlation, and darker red means higher negative genetic correlation. And you see blocks of like the blues here. Okay, so you can see this blue, for example, corresponds to the IBD, Crohn's disease, eczema, celiac. So this corresponds to a cluster of immune diseases. So there's a lot of shared genetics for immune diseases. That's good. And this one has to do with, say, for example, schizophrenia, anxiety disorder, anorexia, major depression, neuroticism. So this actually shows a lot of genetic correlation, positive correlation among the psychiatric disorders. So it's all consistent with what you observe. Now, if you look at the negative correlation ones, right? So there's a cluster of like a sort of a reddish color here. So this corresponds to the negative genetic correlation between these traits. There's insulin, BMI, type 2 diabetes, cardiovascular disease, and height, body weight, of cognitive function, education. So there's a negative correlation here. And also you see the negative correlation here between BMI and the ASD, HDL. So you see this is symmetric here, but also you see negative correlation between education, cognitive function, and neuroticism, major depression. So what this suggests, if someone's genetically more likely to be schizophrenia, bipolar, the cognitive functions tend to be like lower. So this all makes sense. So the next thing is, we spent 5 minutes. I think it's a fun part. So this is about a GWAS study of this million veteran program in the US. So this million veteran program aims to really identify the genetic basis for a lot of things, like affecting the health states of veterans. It's a big study. And so this is a study PIs, Joe Galenter at Yale, and Mer actually co-mentor of time when Tan was at Yale, and Merstin at UCSD. And so just to give you one example, sleep hours. I think as students, I'm not sure here, but in the States, for college students, all lack of sleep. So habitual short sleep duration associated with adverse metabolic, cardiovascular, and inflammatory effects. And also people in the literature estimate the heritability of sleep duration, how long you sleep is genetic, right? Maybe there are some genetics that actually contributes how long you sleep. And from twin and family studies, it's about 20 to 40% actually is due to genetics. Right? And so this actually study gives us some sort of data to really estimate or infer what genes actually may be affecting sleep duration. So it's just very simple question there. How many hours do you usually sleep each day? Right, so you say there's going to be a lot of issues with collecting data this way. There's going to be a lot of errors, but regardless, the veterans were asked to fill in one of these the 6 bubbles, right? 5 or less, 6, 7, 8, 9, 10 or more, right? So you check one, and then that's the data you have. And we have data from a lot of people. So actually, this is a histogram for the sleep durations for European Americans and African Americans. This is actually it's new to me, but actually was actually shown in the literature. This is for African Americans. They seem to sleep less compared to European Americans. And we just focus on those European Americans. So you see the number of the data points we have. So we did the first case control association study. Like you may say, oh, how about just do sleep hour duration, treat it as a continuous variable? Let's say let's do something else. We may lose information here, but we'll see what we get. So we look at the low sleep hours. So we say the 5 hours or less. These are the cases. These are people who say, okay, they have a problem, right? They sleep 5 or less for whatever reasons. Maybe biologically they cannot sleep more. Controls are the 100,000 people here who sleep 6, 8, 9, 10 or more. So these are the controls. So these are very simple first pass analysis, like case control studies, sleep hours, right? We want to see if there's any genetic variants we can identify. So we just did very simple GWAS. Lo and behold, there's only one SNP making the cutoff, right? That's this here. And but you see something else, and this is a QQ plot showing this departure here. And we also use this random effects model and to estimate how many variants, how much genetic variation can be explained by the SNP on the chip based on the model I just described, about 6%. Realizing the questionnaire is very rough. This may have to do with the measurement error for the questionnaire. So what's about this one, this particular gene? So this gene is the RS number, p-value significant. It's close to the CNPM gene. Actually, this region has been implicated for schizophrenia, ALS, autism, anxiety, depression. So the same gene that's actually implied here for low sleep hours actually associated with a lot of other things. But there's only one gene. I hope based on today's the first hour of the last 50 minutes, I'm convinced there's a lot more we can learn by going below this one. We can do a lot more than that. So we just did that. We look at the genetic correlation, right, between low sleep hours and a bunch of other study, other traits. So we saw the negative correlation, genetic correlation, not the observed correlation, but genetic correlation between low sleep hours, age at first birth, cognitive function, education level, bipolar disorder, height. So all these ones are significant ones. These are significant. And this positive correlation between chronotype, that means that you're early morning person or like a late night person, smoking, epilepsy, depressive disorder, neuroticism, etc. Body mass index, right? So you say, if you sleep genetically, if you genetically you tend to sleep less, genetically you tend to have a higher body mass index. Okay. So that's the low sleep hours. I say, okay, I want to do a GWAS for high, long sleep hours. The same data set, but I can look at different slices. And in this case, we say people who sleep 10 hours or more, they are the cases. People who sleep 8 hours or less, they are the controls. So there's 6,000 cases, 131,000 controls, right? So I can do this GWAS. There's nothing significant. Okay, so based on this one. But we can still do genetic correlation because I'm saying there may still be signals here actually by using the tools we develop. We may be able to actually identify some hidden signals here. Look at what we did. Yeah, so we saw negative correlation between age and first birth, positive correlation between long sleep hours genetics and schizophrenia, and the resting positive correlation, negative correlation between resting heart rate and the long sleep hour, positive correlation between depressive symptoms and type 2 diabetes, right? So we find these genes. Sort of there may be some levels of evidence, different levels of evidence, such as certain variants I put people up for to sleep longer hours, people sleep fewer hours, right? And then we can ask the question is, what if look at the genetic correlation between long? For example, you look at the depression symptoms, right? We can calculate the genetics correlation between long sleep hours and depressive symptoms, right? Also, we can calculate genetic correlation between low sleep hours and depressive symptoms. So these are like different genes identified. So what do you see the sign of this one? There's long sleep hours and low sleep hours. What do you see the sign? So for every single trait here, I can calculate the genetic correlation between long sleep hours and this trait and low sleep hours between that trait. So I'm running out of time, but I'll just show. So this is the scatter plot. So overall, you see what? You see a positive correlation there, right? And just side by side, to look at for this one. If you look at low sleep hours, you identify those genetic bases. And the genetic correlation between those and the depressive disorder is positive, right? If I have a genetic variance putting people at higher risk for long sleeping hours, must be the different set of variants, right? But still, they have the positive correlation between depression, right? So these cartoons are summarizes, this short result. Like, it says sleep deprivation is costing Americans millions of dollars. That's funny. Too much sleep costs me money too, right? So you cannot, genetically, when you cannot really force it, when people sleep less or sleep more than typical, actually they have higher risk for like a lot of different issues. Either one would not be a good news. The last 5 minutes, just to, this part is mostly entertaining part, but to show you that there's a way we can really help people to improve their goals. So this is a cover from 2012 in Time, and it's a really like designer baby story. You want to know my future? Essentially, the idea is like people hypothesizing if you really know someone's DNA, you may be able to make a lot of predictions about the future risks for, say, for example, the Alzheimer's, diabetes, etc. Right? And a lot of people in this room may have heard this company on 23andMe. Have you heard about this company? Yeah. And so if I some actual cash, they have a holiday or Christmas sale right now, and they can get 50% off by sending your biological DNA samples in. What they are going to get you? So if you spend $79, and you can tell you, they can tell you ancestry, right? So people in this room, you say, I don't really care. I'm Chinese, but maybe more you can learn. But also in the U.S., people want to learn more interested about ancestry. Also, the DNA relatives funder in terms of 23andMe has a large collection of DNA samples from other people. You may end up finding your twin in that database, right? So maybe yourself or maybe someone else. So that's what you'll get from $79. For $149, in addition to these two, but also you can get you can get yourself assessed for genetic health risk for celiac disease, late-onset Alzheimer disease, Parkinson's disease, cystic fibrosis, sickle cell anemia, so all different things. So that's what they claim you get. Now, if you rush now today, you'll still you can $449 you can get that. And so actually, our group have been really developing some method to help to get better prediction on this. And so these are a couple of papers published by Yiming and others in the lab. The first one is really trying to leverage the functional annotation information. So when we build prediction model, not only we just use different columns, but also we include annotations for different columns. So that's the first paper. The second paper really is saying, in addition to including the annotations, but also we incorporate genetic correlation, right? So if you want to predict someone's disease risk for schizophrenia, but also we know bipolar is a genetic correlation between bipolar and schizophrenia. And the information you get from bipolar GWAS can actually help you to improve prediction for schizophrenia. So this actually is a two paper. When Chan was at Yale, there was a paper written along the similar line, but that require individual genotype data. But this one doesn't. It just requires only summary statistical level data. But I see there's a couple examples here, like this is genetic correlation between the prediction and the real the phenotype. There's a Crohn's disease, celiac, and type 2 diabetes. If we only use the markers themselves without using annotation, that's the correlation we get. If you include annotation information here, actually we can observe some improvement. Saying by incorporating functional annotation there, you can do better genetic risk prediction. And this is using the genetic correlations, Pleiotropy information there, we can also improve that as well. And if you combine both the annotation and Pleiotropy genetic correlations there, you can get more out of it. And this tells you the AUC. So if you know a little bit about AUC, these seem to be large, but they don't look very impressive. So the story is, we can, on the statistical end, we can help people to predict. But on the practical end, you just need to be very, very cautious about what these people claim about their reports. So just to summarize, what have you gone through in the last hour? Just show you that the statistical thinking and approaches can be quite helpful for people to really dig more out of the genome-wide association data instead of just reporting individual p-values. You can learn a lot more about the genetic basis. For example, how many variants are there, and I didn't talk about, and also how, if you could identify all the variants on the chip, how many can explain the phenotype, and also talking about the tissue, the cell type relevance for given disease, as well as the genetic correlations. And the other one is for the example I talk about, to me, is a fun example of the sleep hours. If you just go straight into treating the sleep hours as a quantitative trait, you would miss low and high sleep hours. So this one is saying, you think about the quantitative, you cannot just say, let's do, if you believe dichotomizing quantitative leads to loss of power, that may or may not be the case. In this particular setting, I assume for a lot of like the physiological traits, either too low or too high is not a good thing. So in that case, you probably more care about how to really analyze the GWAS results for those traits. With that, I'll just thank Chan Yang, who started this my talk about this, the genetic, the heritability part when he was at Yale. And Chung Shi really leading the effort to annotate the human genome using different sources, related genome annotations with the GWAS results. He's now at faculty at University of Wisconsin Madison. And the last part I just rushed through. I think it's a really fun part, but we tried very, very hard. We seem to be, we could improve, but only like with marginal, very, very marginal sort of improvement. Yimin Hu, he's a fifth-year student in Biostat at Yale. And many other members in the lab have contributed this work and supported through a lot of different grants. And this NIH, NSF, as well as the Million Veteran Program, this is a small subset of people who have contributed to that project. With that, I'll conclude my talk. Thank you for your attention.
I have a couple questions. First one is I'm very curious about the last part, about the positive correlation between the long sleeping and short sleeping.
Right.
So let's be confused. So like say, if I have one SNP, so this SNP can make me either.
There's a different SNPs. It's a different. Yeah, you identify SNPs putting a higher risk for long sleeping hours, and also have a different set SNP putting a low risk for low sleeping hours.
But if you do the study between the long sleeping and the short sleeping.
We haven't done that. We haven't done that. Yeah. This is still ongoing, but that's work.
Okay, second. So second question about the first part about when you identify a hotspot from the GWAS study. So you mentioned several different numbers. Sometimes the studies quarter million, sometimes several thousand. So what's the red number for different questions? I mean, so what's the perfect number of patients?
For any question, the red number is infinity, right?
Find the right number. Yeah.
There's no right number. It's how much budget you have. Yeah.
Okay, how much? But the result quality will be different, right? So.
Yeah, I think there are people have really worked on how much power you can really improve, how much more accurate you can get. Actually, Chan done some work on this. Along this line, you can really get a curve, in terms of return of the sample size. But it's always the larger sample size, the more accurate you can estimate, the better precision you can get, the more genes you can identify. It's always about the question whether you are willing to put in another $1 million for some marginal return.
Yeah. Okay. So last question about your basic hypothesis. Because from the beginning, you mentioned if you want to find some significant ones, the non-hypothesis is always everything is equivalent or everything is independent. All the samples should be independent. But when you do the detection, let's say you have a million patients or a million people, so you cannot guarantee all the people are not relevant. Actually everybody is related, right? So when you do statistics, so can you consider the correlation in the samples and consider samples are related or family tree or any tree back
Yeah, I think there's a lot of effort going on, and my academic sister, Mary Sarah Mackey, most of her career has been developing tools to do genetic association studies incorporating pedigree structure, either learn from known pedigrees or infer from genetic correlations. There's a whole set of methods. But in this one, we try to remove all these people, those pairs that are potentially related, try to make things easier.
Yeah, but usually it's not possible, right? So even it's not from.
No, you can remove maybe the second, third degree, right? That's possible. You can look at genetic markers. You can pretty much infer, okay, these two people, they are related. But if they're distant related, and you can incorporate that. So there's a degree of, if you have 1 million people there, definitely you'll get something. And the people try to remove some of these clearly related individuals as a simple step. But if you can still incorporate those, but computationally that will be a little bit more complicated, more demanding. But it's still possible to do it. Yeah.
So I also have a couple questions. And say right at the beginning, I'm neither a statistician nor a mathematician, but I am a geneticist. So the first question relates to the sign.
Yeah.
Are the statistics as powerful with negative correlations as with positive correlations? So if, for example, an allele actually lowers the risk, can you see that as clearly and as robustly as you can a positive correlation?
It really depends on the underlying model or underlying mechanisms. If there's some kind of a symmetry there, no, if there is some kind of symmetry in terms of an allele can increase someone's risk for, say, make things easier, height by certain centimeters, and lower by another given level, certain centimeters, and that the test will be equally powered to identify positive correlation or negative correlation. But on the other hand, if you think most of the variants in the genome, there's lots of function variants, other things may be more likely to make things worse or make things better. And then it's not the function, it's not the issue of the method, but more issue of the underlying genetics.
I was thinking more of complex traits like schizophrenia, like Alzheimer's, where it would be hard to say someone got better. If you're normal, you can't be more normal than someone less schizophrenia. So there is no, you could, in that case, you can only see it in one direction, even though a SNP might be correlated with protection against schizophrenia.
I think that you can put it the two sides of the coin, right? In a way, you have two variants, right? There's a protective variant and a risk variant. Right. Yeah. So if you have protective variant, you are more likely to be normal. So that's like you can think about it as the odds ratio there. That really quantifies the effects, right? So that number is we're capturing to calculate the correlation between that. Right. So yeah.
The second question would be what geneticists call metastasis.
Yeah.
So, and you talked about this a bit through covariance approach.
It's not part of the. I think that's really saying our model is very simple and very naive, and what I will suggest, this only suggests that this is suppose this model is correct, right? And this is a heritability that we can, this will estimate the heritability that can be explained. Well, heritability probably not the right term, right? People use that. The variance, the variance of trait that can be explained by the markers on that chip under this model. If there's epistasis, if we could get an epistasis model, our model can do a better job. So this one, I would think about the lower bound for what can be explained. But whether you can, the question is how to really develop an epistatic model that makes sense, right? If you include all the second order interactions, right? That's.
I was just thinking of a very simple model where you go, SNP by SNP,
Yeah.
and even if it has zero effect,
Yeah.
that you reanalyze all the data independently for each type, each
Right.
position. So I'm not being clear. You'd lose statistical power, but you'd run it once with the AA haplotype, once with the AB haplotype, once with the BB haplotype.
I think that's a great suggestion. Yeah, you can suppose you have a major gene like ApoE for Alzheimer, right? You could do a model without the ApoE interaction term, the other one with the ApoE interaction. You can do it, but that's a great suggestion. But yeah, but you cannot get. It's hard to get a model including every possible interaction terms. Yeah, but for major ones, that may be, I think, a great suggestion you have. Yeah, you can get a model that's two sets of terms, additive terms, and interaction between ApoE and everything else. Yeah.
One concern with GWAS is always that the cost effectiveness. You have 100,000 samples, 100,000 markers, and the results are very few reach 10 to the minus 8 or minus 7. feel that something is missing, do you think it's a biology or genetics, or it's a methodology that we can improve?
It seems I failed my talk.
No. You are using function. Yeah, your methods is very good, but still you cannot increase much the output. The GWAS itself gave very few results. Even add on your functional annotation, you cannot increase much. Right?
No, I would say.
So overlap can be even less than the GWAS output, not increase the number.
It really depends on your goal, right? If you want to do the bin counting at the end of the day, yeah, sure, you don't get a lot of the significant results after bufferonic correction, after multiple comparisons. But you still, for example, if you want to analyze or study a functional row of a variant, right? Suppose you get some GWAS, you get something. What kind of experiment you want to do in the lab?
Okay. Functional annotation is very good. I'm talking about the original GWAS.
Right.
They're missing a lot for some other reasons, I think. For example, we assume the biallelic, one allele is protective, the other allele will be risk causing.
Mm-hmm.
But in fact, that may not be the case, because they may just link to the causative, may not be just functional.
I think no one is trying to make a statement about the functional, the causal part. Say we saw association there, and then you need to incorporate.
The objective is to identify the functional.
Yeah, but you have to do it step by step. Right?
So when we do GWAS, we consider not only the allele frequency, we also consider haplotype, right? Do we consider, because everything is generated by recombination. If you have one recombination and another recombination, we'll switch it back. The same allele will come back. So
that's, I think, the functional annotations are probably even more relevant. They say if you have this region, usually you look at these papers, they say we identify loci, not variants, right?
Yeah.
So these loci are associated with the trait. But the question is, it's a, I don't know, not 100,000, maybe $1 million question about which variants in those regions are actually.
Okay.
I mean, even the most recent paper you look at is from the.
Let me rephrase.
Yeah.
If you have the choice of whole genome sequencing and GWAS, which one do you prefer? Do you think sequencing will be better than GWAS?
I think there are people right down. I think that for GWAS, it give you some ballpark estimate. I think for example, if you really want to identify the functional variants, and people may argue probably less will be like the less common rare variants. And but you cannot do the whole genome sequencing for 500,000 people, right? That's going to be even more costly, more difficult to justify. And
okay, come down to the functional annotation.
Yeah.
How much fold can you increase the results? You can save the results, some missing results. You can get.
No, functional annotation is. I mean, you can use that for different purposes. So we had a paper, right, to talk about how much it's going to be case by case, right? So in our paper, we show like if you look at the early phase schizophrenia, and if you incorporate functional annotation, you maybe increase. If you only can afford to do 10 replication studies, you may improve your chance of success, right? That's more for replication studies, but it depends on what question you are asking.
Functional annotation is very good. But you're talking about the overlapping. If the GWAS already missed out completely, not even getting to 10 to the minus 5, minus 6, then you cannot see feedback.
No, but as
But-
what
but just
what I
overlap.
showed, I hope hopefully if you
Yeah.
despite of the lack of any significant results, we were still able to identify some genetic correlations that different people may take on this for different purposes. For example, people like you might have heard a term called the PHEWAS, right? The phenotype association analysis, where you look at variant instead of looking at associate between like one phenotype, a million variants. You look at the associate between one variant, a million phenotypes. And in that regard, if you really have identify some genetic correlations between certain traits, yeah, there's on the global level, and also if you look at the local level, and that may not be very basic research to understand the functional role, how this variant affects the disease risk. That can be very, very tedious, long, like laboratory experiment process. But if you say on the very, very practical end, right? So if people already seen that the benefit, if I have a sort of a treatment for a given disease, and so this kind of analysis, if I can identify or can make statement about there may be some shared mechanism, some pathways between these two diseases, where the second disease doesn't yet have a treatment. And if this is a disease that's hopeless, it may be worthwhile to give you some suggestive treatment pathway to move forward. So there are different ways you can utilize this. And it's just a result, right? They say you can also genetic it's very global level. If you want to understand molecular biology, it doesn't help to understand molecular biology, the basis of the disease. But just gives a way just to how much you will invest hundreds of millions of dollars into this. Why not just to make more out of this, right?
So my point is really the problem with the GWAS. Functional allocation trying to help the GWAS as a problem.
I don't think GWAS is a problem. It's more of how you can make more out of GWAS. It's a challenge, right? It's
I think that GWAS is missing too much. Cannot be just sit back by objectives.
No, but you still need to collect more data. It doesn't mean you can stay with the GWAS and you'll solve the world's problems. But the data are already here. If you really want to design your next phase study, you may have different options, right? It's now saying, let's forget about GWAS. Let's start from scratch, the fresh.
Thanks for the very enlightening talk. So the first question is, when you estimate the genetic correlation, so you have two linear regressions, right? So do you require the X to be the same, or I see you have X and Z?
To be the same, yeah.
You require that.
Because a lot of people, they impute markers on the same reference set.
Okay.
You have the same variance, yeah.
Okay, so the second one is a very naive one. So usually in statistical genomics, people always use false discovery rate, so
right.
you can improve the power. So in GWAS, it seems people still mainly use Gonfaloni correction. So if we, for example, use false discovery rate, it seems, for example, we can cut the threshold at 10 to the -6. Is there any specific reason, or it's just a tradition?
I think if you look at early on, there's a paper by Chiara Sabati many years ago. She proposed, actually wrote a paper about using FDR. And on the practical end, it's basically like Bufferoni. So because the signals are very sparse, very weak, so in that case, the FDR is really going to be a good method if you have a lot of signals, right? And then that can really help in power like microarrays. But for GWAS, when you have very few signals out of millions, it doesn't really make much difference at all. And the second myself is maybe for me more philosophical. I'm not a big fan of FDR, in a way that what really matters is you have all the individual tests, you rank all the things, right? Unless you change the order of these different ranks, you do not change the world in a way. Really, to follow up, whatever people will do will be decided how much they have, how much they can afford.
So your point is when you use functional annotation, you can change the order?
Yeah, that's the point. That's the point. Yeah.
Okay, let's thank the speaker again.
Okay. Thank you for the discussion. Yeah, appreciate it.